All right, our next speaker is Angelea Belfiore. She's an undergraduate from the Mercyhurst University.
Hello, everyone. My name is Angelea Belfiore. I'm a junior environmental science major at Mercyhurst University, and today I will be talking about a concentrated aspect of this big project that we're doing with respect to rotifers and their population dynamics, with respect to Cyanobacteria blooms. So a little bit of background. We didn't really get into the details of this because we didn't, but for my project, or for this specific presentation, I'm talking about rotifers, so here's an introduction to some of the rotifers. In terms of what is in the bay with plankton, phytoplankton or zooplankton, we have the diatoms, green algae, dinoflagellates, etc., which I'll get into later because that will show more of what is important with the results. But with zooplankton, you have grazers, predatory, and so with the focus of this, you have Caratella colaris, Polyarthra, and Caratella quadrata. And so I'm focusing mostly on the middle one, the Polyarthra, but they all play an important role with respect to the total planktonic community. So the parameters of the study, Dr. Diz and Dr. Campbell both kind of talked about it. It's to kind of like predictor model, but I'm not really, that's not seen to fruition yet, so I won't get into it because I'm not really sure of all of the variables that go into it. And so to hone in onto the rotifer life, they are significant with the total planktonic community because of their biomass and their numbers. They are dominant because they are the link between the primary producers, the algae, and the bacteria, which aren't primary producers, and the zooplanktivorous fish, as well as the predatory zooplankton, such as Leptodora, the copepods, Daphnia, etc. So for our sampling location, we also talked about this previously, but Presque Bay, and as it was mentioned, Presque Bay, which is not as similar to Lake Erie, it's much shallower, and therefore has a lot more variability in terms of the temperature from a day-to-day basis. So this is just us out on Dr. Diz's sailboat when we couldn't take the environment one day, but we're collecting just temperature and light readings, and then this is from one of us on the boat, and then these are the 3 sampling locations for which we took our samples. And so some of the methods, and so from last year, from May to October, every 2 weeks we would go out, and then from this year, every 2 weeks from June to September, taking triplicate samples at each of the locations. The first year we took vertical and horizontal tows with the 5-meter plankton, 65 micron mesh, Wisconsin-style plankton net. And this year we decided to do vertical because we saw the results and realized we could hone in on just the vertical to get a better reading of the communities. At each location we would take set sheet depths, light and temperature readings along the water column, and then of course we retrieved the samples. Compared to our sampling last year to this year, we decided to not use a preservative this year than we did last year. So in this picture you can kind of see it. We would go out in the field with our 10% buffered formalin in each of the jars, collect the samples and then put them in there. This year we decided that we didn't really need to do that, and I could process the samples within a week while they were still alive, and that would be that. So in the lab, these are some of our samples from this year, and I'll get into the differences in colors and such. So each sample was enumerated via a Sedgwick rafter chamber, and it was a 1 milliliter sample of each of the 9 samples that were taken per week. they were reduced once all the samples were counted and accounted for. They were reduced using a plankton splitter, one of those things where you turn splits into two. And those were 200 milliliters, and then one sample was preserved with a 95% ethanol alcohol, and this one was 10% buffered formalin. And you can see the difference between the two samples, not only between the May and the August, how much more is just in here compared to into here, but also with the color and the pigment. And that's why we wanted to do, or we wanted to process the samples live to see, just how biology works, how it all moves together. And I think that was probably for the best. And so, with what we saw this year as well as last year were diatoms. So Fragilaria, Asterianella, Melisira, which was the #1 diatom that we saw for green algae, Pediastrum simplex as well as duplex. And then, of course, the cyanobacteria, Microcystis aeruginosa, Lingbia, Limnetica, as well as other species, Anabina spiroides, but also Anabina wisconsin and Anabina floss aquae, and Aphanizomenon. And some of the zooplankton species, we had both cyclopoid copepods and calanoid copepods, Zooplankton, Leptodora, as well as the rotifers, but these are more predatory, so I figured I would include them in. And now we get to the results. Very interesting, very unpredictable because of the variability between the past year and this year. The numbers are kind of all over the place, like just this one goes up with the cyanobacteria, but that was Caratella erlineae, and this year the numbers are very small. Now there's a lot of speculation as to why that could be, as well as why Polyarthra this year are all over the place, and last year they weren't, or they weren't as much right here. So I'll get into, I guess, the reasons and the reasoning for why that is, but just to take in the entire variability, not only of the zooplankton, but also the cyanobacteria. And when I'm talking about the cyanobacteria colonies, it's just all of the totaled of those previous 4 or 5 species that I just discussed. And so I thought it would also be interesting to include the predatory zooplankton that I had mentioned in the other slide. So we have the cyanobacteria again, cyclopoid copepods, Leptodora, and the Zooplankton. And I thought it was important to, of course, include the error bars, but you can see how every increase and decrease, they are almost always together, which is a very interesting, but very, just kind of out of the blue. You're not really sure why yet, why that was happening. But to include the Polyarthra species spiking at 200 right when everything else was decreasing makes you wonder why. So there are a few speculations that came up with. So there could be a relationship between the microcystin levels with the rotifers. So as other large zooplankton such as Daphnia and calanoid copepods have a negative correlation with them, Bosma and other rotifers have a positive correlation. The presence of certain rotifers, as I mentioned in the 2 comparing graphs, their presence last summer compared to this summer could be the result of the presence or absence of the differing levels of the toxins. We're not really sure yet how to monitor that, but we could continue to do lab work in a controlled environment where you're the one, the scientist is the one who's introducing the toxins, the levels, and what species is exposed to what level. But because of the 2-year study, we were able to see from those the numbers were going to be normal every year, or if it was just by chance that there was a spike in a certain population species. And so for further works, these are all very zoomed in pictures of Microcystis aeruginosa, and you can see the little bacteria or little filaments around it stuck in the mucilage, and that raises the question if the rotifers, such as Polyarthra, are feeding on the bacteria, and those are the bacteria surrounding the microcystis, then perhaps their populations are spiking because of the increased amount of bacteria with the clusters of Microcystis aeruginosa. And it also could affect the amount of bacteria could be affected by the stage in which the colonies are either living or starting to decompose. So as they start to decompose, more bacteria are going to be surrounding them, and therefore there'll be more available nutrients for the rotifers, Polyarthra, to be feeding on. So that is just one way in which we can continue this study and continue to look at things. This is just the tip of the iceberg when it comes to the information that we've gathered over the summer. This is just a very specific species that we've looked at, and there's a lot of different outlets we can go to. We took temperature, light readings, and those could play into all of this as well. But because of the time constraints, not only of this speech, but of getting all the information, because our last sampling date was September 21st, there's a lot of information to just kind of digest and see where it all leads to. But there's definitely a lot more work that can be done. So a thank you to the Sea Grant for funding for Mercyhurst University, Dr. Campbell, Dr. Diz, Trevor Sturgeon, who was the man pictured in the first picture with the beard, and to the TREC and the Regional Science Consortium. So are there any questions? Yes.
Are you doing any measures of like something like heterotrophic plate counts or something like that that might help you get at the question of whether that kind of primary production of bacteria is related to the spikes you're seeing in some of the grazers?
We haven't done any of that. This is just kind of looking at a more like larger planktonic communities, so like the algae and the zooplankton, but nothing lower than like the bacteria, because that's just, I guess, not within the scope of the project. But definitely something to consider. Yes.
Angelea, with the, since you have live samples that you can look at, you can see those rotifers in there spinning away, doing their little thing.
Right, yes.
Can you actually observe them feeding?
Well, what's interesting is with the rotifers, they are always surrounding themselves around, a bundle of something, and they're moving through and kind of like grazing over it, which is what they do, they're grazers. But with there's a lot going on within the sample, so it's hard to focus on just one small individual organism when there's a giant copepod swimming by and moving everything, But it's definitely something that you notice, because those are the ones that, are swimming, but the rotifers are the ones that are honed in on a certain clump of something and are focusing on that area.
But you're pretty sure that they're not feeding on the microcystis?
Oh, no, they're not feeding on like the actual microcystis cells, but perhaps what's around them in like the mucilage.
It's way too much of a mouthful for them.
Yes.
I know you mentioned this, but I'm just thinking about, how did you sample? Was it vertical or horizontal?
Vertical.
Okay.
Yeah. The first year we did vertical and horizontal, and then we did some testing, and we realized, like with the numbers that we got, we realized there was no significant difference between the two. So we realized with a vertical toe, we would be getting a better, I guess, representation of the whole water column, and like instead of just the horizontal aspect of the first meter of the water.
I think there's one more question in the back. Does somebody have their hand up back there? All right. Thank you very much, Angela.